So recluse spiders, genus Laxosceles, also known as brown spiders or fiddleback spiders, and they're, because of this fiddle shape on the cephalothorax, and there are about 8,000 cases a year in Brazil alone. About 10% of their bites cause these dermonecrotic lesions that may require skin grafts or then be disfiguring. And about 1% can become systemic, which can lead to hemolysis, damage to kidney cells, leading to renal failure and even death. You can see some of these necrotic lesions here. However, there's no diagnostic test, and there are no directed therapeutics yet. And the antivenom is not widely available, and it's only moderately effective. So clearly some new therapeutics are needed. However, here we go. It's not yet fully understood how it binds, so I'll talk a little bit about that. So this is the current range of the Sicariid spiders. That's a family, including Laxosceles. Notice here that they have a primarily tropical or subtropical distribution, including Laxosceles reclusa, which is the infamous brown recluse, but there are also many species in South America and up in the Mediterranean and parts of Africa, which is Sicarius, which is a sister genus. However, this is the predicted future range of what's considered one of the most invasive spiders, Laxosceles rufescens, originally from the Mediterranean, but by modeling, which takes into account climate change in the future, it's predicted. Notice here, it's highly predicted to have a population established in China, the most populous countries in the world, and parts of Australia. In fact, there's already been a small population established outside Adelaide in Australia. So this is predicted to become a global problem. So the toxin responsible for those effects is a phospholipase D, shown here modeled on the surface of a membrane with a lipid substrate docked in the active site. And what happens is this toxin comes and it sits down on the surface of membranes. It brings up that phospholipid up into the active site. And then it catalyzes this really unique cyclization reaction where there's acid-base catalysis with these histidines in the active site, and it results in this intramolecular cyclization of phosphate, resulting in this very strange cyclic sphingolipid product, cyclic ceramide phosphate in this case, with release of that head group. Now, despite very convincing NMR evidence by members of my group, it's still highly contested in the field that this is in fact the product, because there's no direct structural evidence of this lipid. However, it's thought to play a major role in the membrane destruction that's seen here in this case. Phospholipase D was added to micelles or vesicles, and you can see there's this membrane disruption. And this toxin, when produced recombinantly or isolated from crude venom, you put it into in vivo or in vitro models, and it recapitulates the whole syndrome, dermonecrosis and hemolysis. So this is which is unusual for spider venoms, this is the enzymatic active toxin. So I solved the structure of phospholipase D my goal was to get it bound to substrate and product. So to understand how it interacts with substrate. Amazingly, it's bound to 3 lipids simultaneously in the structure. So here, this is that cyclic product bound in the active site. And then there are 2 substrate molecules that were bound in allosteric or non-catalytic sites, which is very interesting and surprising. But note, now we have very direct structural evidence that this is in fact the product, is a strange cyclic lipid. So this was the previously proposed catalytic mechanism based on in silico docking. Just note here that there's a head group binding, we have these aspartates mainly coordinating this magnesium that coordinates the phosphate of that lipid. And pay attention that there are these histidines that are poised for the acid-base catalysis. So there was a second structure I solved with substrate bound in the active site at a bit lower resolution. But when we compare these two side by side, this is from the structures. Here you can see the product in the active site coordinated by the magnesium. But when we look at where those residues are, they're perfectly poised for this acid-base catalysis, resulting in this captured product in the structure, aligning with that. So that confirms that originally proposed mechanism. Now, very surprisingly, when we look at the asymmetric unit and then related by crystallographic symmetry, it actually forms this homotetramer with these. This is the asymmetric unit. It has two of the enzymes, and then related by symmetry, there's two more, and they sort of form this almost micelle-like superstructure with the 3 lipids from each structure in the center, surrounded by the 4 subunits of protein. And this is rare for X-ray crystallography. Structural biologists will know, because lipids, in part because lipids are very flexible. So that was very surprising, and we began to wonder, what does this tell us, if anything, about the membrane interface, since we're forming sort of a micelle-like structure in the center. And this was from some molecular dynamics done by collaborators previously. And the 2 of those loops, they embed into the membrane. And here is from those results showing how the depth of these 2 loops. And when we look at the contacts of those 2 loops that are contacting the membrane, and we look at the structure, highlighted there in purple are the residues contacting the lipid chains in the structure, and they correlate very well with the ones predicted to interact with the membrane, which is interesting. So the structure also explains some findings previously in the literature, which it was looking at the substrate preference depending on how many carbons you have in the lipid chain. And so here, start with, as you decrease the number of carbons in the lipid chain, the max rate here, this was a fluorescence intensity assay, the maximum rate, the preferred substrate has 6 carbons, which isn't a natural mammalian lipid chain length. But when we look at the structure, this is the electron density in the structure, and this means there's high consensus up until about carbon number 6 in the chains. So what this means is that high consensus is that those carbons aren't moving a lot, and so they're likely bound. And so when we look at the residues in the protein that make contacts with those lipid chains, those also make contacts out into about the 6th carbon. And so what that means is that, like, the enzyme binds the lipid chains out until about the 6th carbon. If you have fewer carbons than that, you start losing activity because you've lost contacts. If you have more carbons than that, we think that means it's harder to extract from the lipid bilayer, because the lipid's making more contacts in the membrane. One of the other surprising findings that's quite cool is that here in orange is there was an original structure without lipid bound. And then here in tan is showing the new structure with lipid bound. So these are those 2 loops, the catalytic loop and flexible loop, we call them. And just note here that there is a shift. The other one superimposed here on the bound structure is a previously solved structure of a different closely related enzyme that we think is in an active conformation. And in fact, here's the non-catalytic substrate molecule on the opposite side of the catalytic loop, and it clashes when we map it onto the unbound lipid form. But when we look at the bound form, that loop has moved over. This is the catalytic loop. It's been pushed over toward the active site where in this case, product is bound. And so you have this non-catalytic lipid bound in that pushes the catalytic loop over to essentially assist in the catalysis in the active site. And we have seen bits of allostery in these enzymes that would explain that. So this crystal structure, it confirms the cyclic product very beautifully and how it is converted from substrate. I think it provides insight into how the enzyme actually interacts with the membrane. And because now we understand how it binds to product and substrate, it gives us new information for developing therapeutics. And so we're now pursuing 3 different avenues here for therapeutics. There's a classical medicinal chemistry approach, elaborating on the cyclic product. It's a very unique bit of chemistry, and so we hope it'll provide the selectivity of the therapeutic. We've already generated sequences with the collaborator in silico for peptides that should bind the active site, and we will test those. And excitingly, this week, sequencing should come back from a very interesting phage display library of these bismuth cyclized peptides that have these regularly interspaced cysteines. You just add bismuth and then it spontaneously forms a cyclic peptide, which increases stability. And so we've done a binding screen with that, 4 rounds of phage display. And I'd like to thank the structural work was done at University of Arizona with Matt Cordes and our crystallographer, Bill Montfort. A lot of the evolutionary work supporting this was with Greta Binford. And I'd just like to thank a lot of the collaborators at Australian National University, as well as at University of Queensland, where I'm now working, and of course our funding. Thank you very much.